To the last session today. This is another session on companies. I'll hedge on using the term startup because in contrast to the early stage companies that was the focus of discussion this morning, we chose two talks from later stage companies that came out of universities but have gotten a lot of traction, a lot of financial support, and have created significant handoffs and gotten significant investments. But nonetheless, the technology came out of universities. So really, I wanted the speakers to speak about the stress points, the key decisions, handoffs, and also think about some of the same issues as were discussed this morning. How does one balance their academic life with their company life, with before and after a handoff? And both of these technologies are in the cancer space, and as best as possible, I know the investigators aren't necessarily in the regenerative medicine space, but as best as possible, talk about what components of their story transfers to other spaces, particularly regenerative medicine space, and what might just still be specific to the cancer space. So the flow for this afternoon is we'll have two 35-minute talks and then a combined question and answer and discussion period for another 35 minutes or so at the end. So it's a pleasure to introduce the first speaker, Dr. Thomas Chen. He's a professor of neurosurgery, pathology, and orthopedic surgery at USC. He got his MD at UCSF, did a neurosurgery residency at USC, then got a PhD in tumor immunology, also from USC, and finally finished with a fellowship in spine surgery at Wisconsin. In addition to his academic portfolio and surgical expertise, performing both spine and brain surgeries, he's the founder and CSO of NeoOnc, which focuses on developing and commercializing pharma-based therapies and drug delivery methods for CNS diseases. NeoOnc has significant holdings. It is multi-phase 2 clinical stage biopharmaceutical ventures and has pioneered CNS therapies for cancers in that space and other malignancies. So it's a pleasure to introduce Dr. Chen as he'll talk about his story in navigating out of the academic lab, out of his clinical practice into very substantial in human work.
So I want to thank you for the opportunity to come here and speak, and to Uttam Sinha, who invited me. What I'm going to talk to you today is basically what we're doing as a way of delivering drugs to the brain. And in the course of delivering drugs to the brain, we found that this intranasal delivery route is actually good for delivering drugs to the spinal cord. And so I'm going to talk, first of all, I'm going to talk to you about what we have done. And then I'm going to talk to you about a little bit of the science in terms of what we have developed. So I'm going to start off with talking about starting a startup and how we got started. With the initial idea and the company formation. I'm going to talk to you about some of the grants that have been helpful to us, especially NIH, STTR, and SBIR grants. Fundraising, going from founders to friends and family, Public listing, we actually went public through something called direct listing, not the usual traditional IPO. And then I'll talk to you about some of the clinical trials that we currently have in process. So here's the thing is that, in life, you meet people, things happen, sometimes by accident, sometimes by design. But I gotta tell you the truth that a lot of what happened with my company was sort of like by accident. And so I don't want to be like claiming a whole bunch of things like I did this, I did that, but I'm going to tell you the truth in terms of what happened. So what happened was that we started working on a monoterpene. It's called para-alcohol. And para-alcohol is basically a plant-based drug. It was actually discovered at University of Wisconsin by a biochemist by the name of Michael Gold. And Michael Gold at that time was looking at cell cycle inhibition. And he found that para-alcohol, this monoterpene, actually was pretty good at cell cycle inhibition. And so he took that drug and they were giving it to the patients orally. Okay, and it was treated for patients with metastatic breast cancer, colon cancer. And these patients were taking like grams of this stuff. And some of these patients got a little bit better, but they have one very bad side effect. It was severe diarrhea. And anyway, so they ran up to phase 2. They stopped after phase 2 because most importantly, they weren't getting like gangbuster results with it. It was just, patients are luck. So he stopped doing it. So this man, Clovis Fonseca, he's from Brazil. He works at a university in Brazil. He's a neurosurgeon. And Clovis goes to visit Michael Gold. Now, I can't remember why he went to visit Michael Gold, but he went to visit Michael Gold. So at that time, there was no TSA or anything. He said he went to visit Michael Gold, and Michael said, "Hey, I've got this drug. It's a cell cycle inhibitor." And so he gave him a jug. He gave him a jug of this stuff, and he just carried it back to Brazil. And the Brazilian IRB at that time was not very stringent, and so he was treating glioblastoma patients, and he thought, hey, what? And I should also say at that time this whole concept of nasal brain delivery was getting developed in animal models. So he was thinking, well, okay, so if these patients have all this diarrhea, could I give it intranasally? And could I have the effects of a cell cycle inhibition on these brain cancer patients without the side effects of the diarrhea? And that's what he did. He started giving these patients intranasal para-alcohol. And lo and behold, some of his patients actually had a great response. He had like pictures that show that these tumors actually got smaller. So what happened was that he gave a presentation at the Brazilian Neurosurgical Society, and he got first prize for it. And the first prize was that you can do a little sabbatical in any lab within means, but in any lab that you choose. Okay, and so that's how he came to meet me, and he met me, and he was like all excited about his results, and I took a look at his results and I said, wow, that looks pretty good. But the thing is that, it was so good, how come the whole world isn't using it, something like this? So I told him that, hey, what we should do is that we should do a study where we can run in some academic centers here in the States and validate your results. Okay, so that's how we got started the idea. Now the thing was that at that time I was thinking that, well, the para-alcohol that he was using, you could buy at Sigma Chemicals. Okay, so there was very little IP behind it, all right? And then I thought that, in terms of what you're doing and validating this result, we need to have, if we want to raise money, we need to have some patents behind it, and we need to have a company, because there was no way that we're going to get, write NIH grants enough to make it happen in a quick time. And so that's how we started off with the company, was that we formed a little company. I wrote a patent, and our patent was not anything spectacular or whatnot. What we did was that we took the para-alcohol chemical and we purified it, added a different step to it, and made it under GMP conditions, with the idea that we were going to administer the drug to the patient with a recurrent glioblastoma. And so when I did that, I knew that. So at that time I had written a patent on this compound, and we call it Neo 100. And then we basically wrote an IND for submission on this new compound. And before we wrote an IND, we had received a fast track approval and orphan drug status for this drug. And we wrote an IND, but I didn't have any money. So with an IND, you want to present some toxicity data from large and small animals, and you want to have a clinical trial. So we knew we want to run it like a phase 1 clinical trial, so that was easy. But in terms of the large and small animal studies, we really didn't have enough money to do that at Charles Rivers or something. And so what we did was that we took the patient data from Brazil, and we used that for our toxicity data that we presented to the FDA. Okay, so when I did that with the IND, I wasn't sure what the response would be, because I had never presented an IND to the FDA before. Okay, but you know that when you submit an IND, you have 30 days, okay? And if the FDA doesn't come back to you and say anything to you in 30 days, that was assumed that was a go, all right? And so we submitted it. 30 days passed, nobody said anything. So we were a go. And so we were a go with our phase 1 clinical trial. And that's how it started. Now, I want to tell you, just along the way, that, like I said, that we took advantage of the STTR, SBIR mechanism. Because, and this was good because the funding for a small business NIH grant was much better than for a regular R01 or R21. With the STTR, you basically, the money comes to the company, you can subcontract to the university, and it was a 60% university, 40% company. With the SBIR, it's the reverse, the 60% company, 40% university. That's why we used the STTR. But we did use NIH funding for a lot of our work, and in terms of raising money. Now, fundraising for us came from friends and family, and raising money. Raising money is really funny because it can be like, you can get money and you're like wondering, how did I get money? And then you can make pitches and you never get any money. So it was if I didn't have to raise money, I would be so much happier. But so I'll give you an example. I went out to dinner with a friend, and he actually is a wealthy friend. He owns some hospitals, And so I went out to dinner with him, and he's like, okay, so what are you doing these days? And I said, well, we're trying to do this company, et cetera, et cetera. And so he goes to me and he goes, I like to be involved. And I said, that's fine. And so, doctors, they don't invest more than like $50,000 would be a lot of money for a doctor, right? So he goes to me, he goes, would 2 million be enough? And so I raised $2 million just from that dinner. And then there's other times when I'm like giving pitches and all this stuff and nothing ever comes through. So what can I say? It's a numbers game. It's a numbers game for raising money. And you can say, well, I'm going to, prepare this, I'm going to prepare that. And I've certainly given pitches to, venture capital that as soon as you start talking, their mind is off somewhere else. And they never, understood what you're saying anyway. And then there's times when it happens. But I basically raised about $10 million over a course of about 8 years to fund the company. So these years, so we basically started work on this in 2015. And so most of the fundraising actually started 2019. So basically, I had a very dry spell for a long time, and I had that very dry spell because somebody introduced me to his brother. A friend introduced me to his brother-in-law. And his brother-in-law used to be a banker at Citibank. Okay, so I'm like thinking very naively, okay, banker should be able to give me money, all right? So I said, and he said, and his brother-in-law said, he would make a good CEO for you. And I said, okay, fine, I'm gonna make him my CEO. And then I discovered the truth about bankers. You know what the truth about bankers is? Bankers take in money, okay, and they use the money, right? But they don't know how to raise money. They can't raise money for their life, and so my banker CEO never raised me any money, and I actually fired him because what happened was that he said we need to have a financial advisory board, and so he invited some of his friends that were financial advisors. And then what happened was that one of his friends actually decided to invest $50,000 into our company. Okay, and we were like running on fumes for money, and I'm like going, $50,000, where is it? How come we're not spending it? So my CEO took in $50,000, but he didn't want to spend the $50,000 because he didn't want to lose his friend's money. And I said, well, how can you be a CEO of a company when you have so little faith in the company that you're afraid of losing your friend's money? So I got rid of him. And then when I got rid of him, then I became the CEO and I started raising money. And that's how we got started. And so what happened next was that, again, it's all by chance, okay? I was pitching the company to somebody that there's a fundraiser called introduced. They basically called me up and said that, hey, I'm interested in introducing you to this guy who looked at your portfolio and was interested in the company. Okay. At that time, I was actually pitching for another company because I was working on two companies. One was this company and one was a company called Disintegrins. And so I gave this particular person a pitch on my Disintegrin portfolio. And then he invited me out to dinner, we talked, etc. And then in closing, I said that, I want to be in all transparency. I want to let you know that I also have a company called Neonc that we're hoping to take public. So this guy, as soon as he heard public, it was like, wow, this was so much more interest to him. And so that's how I got him to be involved with Neonc. And the thing is that he has taken previous companies' IPOs, so he has the bandwidth and the knowledge to take a company public. Okay, if you were to ask me to take a company public, we would not be public today. All right, I mean, I don't have that skill set. I don't have that connections and the ability to take that that company to a public stage. So again, like I said, that in life, many things happen by chance or by accident. I don't know what it is, but we were prepared for something to happen. But it just, I just happened to meet the right person that was interested in the company and wanted to take it public. And that's how we became public. And even when we became public, we were initially thinking we were going to do an IPO. And at that time, when we were thinking about doing IPO, he has a friend who is the CEO of a large pediatric hospital called Sidra in Qatar. And so the idea was that we would get money from Qatar to boost us up for our NASDAQ listing. So I went to Qatar 3 times, okay? And if you ever dealt with Arabs and the Middle East, it's like there's no time schedule. It's like you show up, and when somebody's ready to see you, you go and see them. Okay, and so we were supposed to see the Queen. Okay, the Queen was going to give us a whole boatload of money, and so we went over there to meet the Queen. I actually met the Queen. And the Queen said, the first question the Queen asked me was that, are you going to cure brain cancer? Okay, and I said, I should have said yes. Maybe I would have got money, but I was stupid. I said, well, we're going to make people live longer with brain cancer. And she says, oh, okay. And then at the end of the talk, she goes to me again, are you going to cure brain cancer? Anyway, so the bottom line was that we went to Qatar 3 times. We were supposed to get a $50 million investment from the Qatarians. It never happened. And so during this time that we were going IPO, we were, boasting to our investors that, hey, the Qatarians are coming in with 50 million, you can put in this amount, and we make you guys, everybody make a boatload of money. And then the Qatarians never came. And so our IPO thrust just went deflated. And that's when, so for a year, we basically had a failed IPO. We just took it off the thing. And, before you do an IPO, you have to do the S1, and you have to do a whole bunch of stuff, pay a whole bunch of lawyers. And so at that time, then we decided that, well, maybe what we would do is a direct IPO. And with a direct IPO, what you're doing is basically like if you're selling a house, you can do it by owner, or you can do it by realtor. And so when you do it with the IPO, you're basically doing it like by realtor. Direct listing, you're doing it like by owner. So you collect your own money, you need to have a certain number of investors that are part of this, and then you can list. And so we want to list in NASDAQ Global, and so we needed to have 240 investors involved, and we needed to have $10 million. And again, like I said, it's all by like chance. So we're trying to raise $10 million to go direct listing. And then I had Thanksgiving dinner with an old childhood friend, And he is hemiparetic from a softball accident as a child. But he did one thing right. He bought Apple stock from the very beginning, and he just really believed in Steve Jobs. So he bought Apple stock, Apple stock, Apple stock, and by the time he's our age, then he was worth a couple $100 million. Okay, so I invited him to our house for Thanksgiving dinner. I wasn't thinking about like having him put in money or anything. I just invited him over. And so after Thanksgiving dinner, he said, I really want to invest in your company. And again, I was thinking, oh, maybe put in a couple of $100,000 because he's a rich guy. But he ended up putting in $7 million. Like, again, it's like, where you come up with $7 million pitching? And he put in $7 million, and we had other investors in, and that's how we went public. And again, like I'm just saying this, that everything in life is just like, if you're prepared and you're thinking, you can take advantage of something that drops on you. But it's sort of like, you can't just sort of like plan, so you're going to do this, and you're going to hit this, because it usually doesn't happen. And so that's how we went public. So what I want to do now is talk to you a little bit about what we did when we went public. Okay. Before we went public, we had already done a phase 1 clinical trial. And so when we went public, we had more capital, and we end up doing 4 clinical trials. And so very quickly, how much time do I have? Like 13 minutes. Okay, so I'm going to go very quickly in terms of what we're doing with the intranasal delivery, so everybody has an idea. So this is something about, I'm going to talk a little bit about the mechanism, the distribution, what the particle size is for delivery, what we use for intranasal delivery, and some of the clinical trials. So with intranasal brain delivery, what we're trying to do is we're using cranial nerve to brain delivery. And so it's the 1st and the 5th cranial nerve. We're not trying to cross the blood-brain barrier, we're bypassing it. And so it's a non-invasive way and you're able to do this therapy at home. And the clinical trials are performed in adults, and we also had an IND for pediatric midline gliomas. And this is the way it does for intranasal trial. What you're doing is that you're delivering the drug intranasally. It's going through the nerves, through the cribriform plate, and you're going to olfactory bulb, olfactory tract. And what you're doing is that you're using that cranial nerve, and then that drug that's delivered gets ultimately absorbed by your lymphatic system, or really it's the CSF, and that's how it circulates throughout the rest of the brain. And so this is our drug. It's called NEO-100, and like I said, that we use the para-alcohol, we purified it, and then we made under GMP conditions, and we received the FDA approval for IND, and was fast-tracked an orphan drug status. And these are some dissections that one of our research scientists did. She did some dissections showing the trigeminal nerve and the olfactory nerve, and this is in mice. And what she did here was that she took essentially, she took microRNA, she labeled it, and this is in the trigeminal nerve after intranasal administration. And this is just to show you that you can see the microRNA and its label within the nerve itself. Now, in terms of how the drug is distributed, well, we had the benefit of just using rodents, but what we could do was that we could sacrifice the mouse and then be able to slice it up and then look at the distribution of our drug within the brain. And we found that it was actually pretty well distributed throughout. And what we could also do is that we had a patent where we said, well, what if you took a drug that could normally not cross the blood-brain barrier, but if you could take our drug, dissolve it, and then sniff it up, could you then get it across the blood-brain barrier into the rest of the brain? And so that's one of our patents. We took a drug called Velcade, which is a proteasome inhibitor. It doesn't cross the blood-brain barrier, but if we use it in terms of the intranasal route, it was actually very good. And here what we did was that we demonstrated that the bortezomib concentration in the tumor is 100 times greater via intranasal versus intravenous. And then the mice that had NEO-100 intranasally had a much better survival because the drug actually got to the target. In terms of the distribution of intranasal delivery, I think this is something that is going to be very interesting and new, because what we found was that I initially thought that because the trigeminal nucleus goes down to the cervical cord, I thought that, hey, if we gave something intranasal, we should be able to see distribution down to the cervical cord. And when we sliced the sections, we actually found that to be true. But what we then found was that we actually found the microRNA in the thoracic cord, and it's also in the lumbar. And so now you have a way of, because we currently do not have any great ways of treating diseases of the spinal cord. So this is a way, I think basically it's the same idea. It's really the CSF that's doing the circulation and getting our microRNA down to the whole spinal cord. So this is one, so what I want to do is I want to develop something called basically intranasal brain to spinal cord delivery. And I think that would be a very useful thing in the future. And then for particle delivery, you need to have a certain size, certain delivery. The angle has to be a certain way. And right now we're using something that's pretty basic. And that's because we're using the same idea as in Brazil. Essentially, that is a nasal nebulizer. And so the patient basically has this nebulizer. It's above the mouth, and so they're breathing in, and they essentially breathe in treatment 4 times a day. So very quickly, I just want to tell you about one of our clinical trials. So like I said, we went through phase 1 already, and now we're in phase 2A. The phase 2A, we have to do total 25 patients, and we've enrolled 24. And this is for basically patients with recurrent grade 3 and grade 4 astrocytomas. And we actually found that there's a good biomarker called IDH1 that we're using. And then we have a trial now for meningiomas, and we have another drug called NEO-212 that we're using for all brain tumors, and it's given orally. NEO-212 is interesting because we took the temozolomide, our standard care drug, and we chemically conjugated it to our para-alcohol NEO-100. And so with that conjugation, we form a new compound. And that's undergoing currently a phase 1 trial. And so when we did our NEO-100 phase 1 clinical trial, what we found was that our overall survival rate for this intranasal delivery at 50% survival was about 16 months. And you would expect, recurrent disease to be about 6 to 8 months. So it was much better than what we expected. And then when we did analyze these patients, we found that if they completed more than 5 cycles, they did very well. And if they have this mutation called IDH1, then they did the best. And then our drug was better than standard care for recurrent high-grade gliomas. Our drug basically required no surgery, the bone marrow is not affected, normal wound healing, no radiation. And the most important thing is quality of life is very high because there's almost no side effects with it. And then our survival time was good because if you see that yellow line, that's what you expect for 6 months. And this is what we were getting for overall survival. And right now we have 25 patients enrolled, no significant side effects, good quality of life. And we're not done collecting all the data yet, but of the data that has had with our 6 months progression-free survival, we're about 44%, and historical with the IDH1 mutations, 21 to 31%. And here it is in a swimmer's chart. So overall, we were getting progression-free survival. 44% of patients achieved 6 months. 21% of the patients actually have radiographic remission, and long-term survival about 33%. And so here's an example of a patient that has done very well. You see the circle, that's her glioma. So she's a grade 4 IDH1 mutant. And what happened to her was that she had radiation, chemotherapy, and then she had 2 craniotomies. And then she went on our trial. And you can see that from January to October, she has had almost complete remission without any other treatment with our inhalation treatment. And here I have a little presentation. Can you press the start button there? There you go. Is there volume that you can put up? Is there volume? Can you put up some volume? Sound? No sound? Okay. Well, anyway, so this is the same patient, that MRI scan I'm showing you. And now she's been on treatment now for about almost 3 years. Yeah. No, this is a grade 4 glioma because it's an IDH1 mutation. But she basically has a normal lifestyle. Her husband's a doctor, and so actually I was on I was in Florida. What's that guy's name? Doctor. He was in Los Angeles. I'm just blanking his name. What? Anyway, so I was in Florida doing this show, and we invited her. Okay, next one. Dr. Drew, I'm sorry. Do you guys know Dr. Drew? What? Yeah, yeah. So anyway, so Dr. Drew has a show called Health Uncensored. So I was in Florida with Dr. Drew, and the patient was there. So anyway, next one. No, just show the next slide. Anyway, that's fine. I don't need any more. So I can expand it. Yeah, you're right. I'm sorry. So anyway, so this is just future directions, but my whole thing is that I think that we're on the verge of discovering a new way of getting to the brain and spinal cord. So I'm doing the worst of the worst, which is and it's my field anyway, the neuro-oncology. But later on, we could be looking at neurodegenerative diseases, infections, treated by this matter, and we actually have a number of publications already on spinal cord injury using an intranasal route. So I want to, like, for instance, take, you know how you get a spinal cord injury, you're like it takes you like 5 hours to go like from Good Sam to USC or something. But I want it to be done in the ambulance so you can have intranasal treatment. And then we want to deliver different compounds like microRNA, liposomes, nanoparticles. Anyway, so this is it. I just want to let you know that, you're not dealing with some brilliant business person that is made it all the way to the NASDAQ. I mean, my friend Uttam here would like you all to believe that's to be the case, but it was, to be honest with you, I had the idea of starting something, and it took me 13 years to get to the NASDAQ. But I just kept plugging at it, and to be honest with you, we ended up generating 178 patents. And this was all, some of these, every time you go to a foreign country, you got to spin off another patent, and it's just costing you more money. But we end up having a patent portfolio of 178 patents. And so now what we're doing is, we're visiting the Arabs again. But this time it's through Abu Dhabi. And so the Arabs have this thing called with Cleveland Clinic Abu Dhabi. They're supposed to give us money, so I'm always like looking for money. It's like horrible. Yes, that's what I need to do. So it's the thing. So here's the thing: is that I'm always looking for money, and now I have to add it the sin of looking at the stock price. So anyway, it's not. I don't know that going public is really for me, but that's my story.
Negotiate that. Did you stay an academic the whole time? Because it sounds like a full-time job, but it's 2 full-time jobs. Were you doing?
I have 3 full-time jobs.
How hard was that?
How hard is that? Well, I mean, I multitask. Yeah, but the thing is that I wanted to, here's the thing is that, it took me a long time to learn to be able to do neurosurgery. So I just didn't, as long as I'm helpful to people, I want to continue operating.
I have a question.
We're going to have Q&A after the second talk, okay?
Oh,
Should I take a seat or should I go back?
You can have a seat, so the slides are up. Yes, we'll have about 35, 40 minutes of discussion and Q&A for both speakers after the second talk. So it's a pleasure to introduce Dr. Zhen Zhu, the Li Kai Shing Endowed Professor of Biomedical Engineering at Michigan, also a professor of radiology and neurosurgery. Zhen's research focuses on ultrasound therapy and imaging, and she's a co-inventor and really one of the pioneers and world leaders in histotripsy. She co-founded Histosonics, a company that commercialized histotripsy technology for non-invasive cancer treatments. And a variety of cancers, also neurological and cardiovascular applications. So as co-founder of Histosonics, her work led to the FDA approval of non-invasive histotripsy for the treatment of liver tumors. And to date, this technology has been used in over 2,500 patients in the US and around the world. There's also ongoing clinicals for the treatment of renal and pancreatic tumors. A couple of months ago, Histosonics was acquired for over $2 billion, making it the second largest VC-backed medical device transaction ever. As a testament to Zhen's technology, she's been elected a fellow of the National Academy Inventors, as well as AMBI and IEEE. So unfortunately, Zhen couldn't be here in person, but she'll deliver her talk virtually, then stick around for the Q&A. So it's a pleasure to introduce Zhen Zhu, our second speaker this afternoon.
Thank you, Dave. I actually, it doesn't allow me to share video. Can someone allow me to share the video so I can turn on my video? It says it is disabled by the host. Oh, I can just share my slides. Okay. I will keep trying of sharing the video. I think it will be helpful for at least a panel discussion. But thank you. I apologize not being able to be there in person, but thank Dave for the invitation and me being able to share my experience. So Dave asked me, when he asked me to give this talk, he said, share the mistakes you have made and the lessons you have learned through your journey of funding and Histosonics and taking get to the FDA approval. And I thought, gosh, I have so many of the lessons I've learned. I have made so many mistakes. So I have plenty to say. So I said yes. And I can't say no to Dave also. So I want to share with you, this is actually has been a 25-year journey. First disclosure, I have, of course, conflict of interest with Histosonics. So I want to share with you my 25-year journey from invention of histotripsy, then all the way taking it to FDA approval, as well as the whole entrepreneurship journey. So first, what is histotripsy? So the idea is using ultrasound device placed outside the body and generate bubbles. And that bubbles destroy the target tissue. So it can be a tumor or can be any pathological tissue target, but without incision, without radiation, without injury, without pain, and we can remove the tumor. So that's the idea. And the reason it's called histotripsy because when my co-inventors and I coined the term histotripsy, we follow the rule that all medical terms is based in Greek. So histo means soft tissue in Greek, and tripsy meaning mechanical breakdown. So the idea is to actually invent a first non-invasive technology to perform a real surgery that can actually mechanically remove tissue, just like a surgery does. So this is the histotripsy clinical device called the Edison device that's manufactured by Histosonics. It's got the FDA approval in 2022 for non-invasive treatment of liver tumors and used clinically. And what it is that you have ultrasound device. So this is an ultrasound device to generate histotripsy with an ultrasound imaging probe in the middle, so we can actually see what's going on. So it's an image-guided treatment. And then there's a robot arm here. And the robot arm is used to actually precisely move the ultrasound focus to cover the tumor. And how the treatment goes is at the beginning, the clinician will use ultrasound imaging to image the tumor. And they can be co-registered with MRI or CT scan taken prior to the treatment. And then the clinician will outline the tumor based on the ultrasound image, and the treatment software will create a prescribed pathway to cover the entire tumor with a margin that outlined by the clinician. And then the treatment itself is automated. So this is just a really short video of the treatment. What you will see here is this is a liver tumor segment that embedded in the gel. And then there's a targeting process. Then this is a scan path that generated by the treatment software to cover the tumor. And the treatment really is automated, just scan through the prescribed scan path. And you can see the tumor fragment is quickly liquefied by histotripsy. And after treatment, when the operator cut through, and what you will see is the liquefied acellular debris just leak out. So those are just acellular debris. There's no cellular components in it, and it will be reabsorbed by the body over the metabolism process. And you can tell that the gel surrounding it actually very fragile, but it's not damaged because it's a non-invasive procedure. So what are the clinical benefits of histotripsy? One is non-invasive. That's the kind of the major strength. This is a patient with a central metastatic colorectal tumor in the liver, and the surgical operation will really damage the overlying liver tissue. But this patient received histotripsy. As you can see, that liquefied acellular debris absorbed by the body. And within 30 days, I already have substantial reduction of the tumor volume, and this will just absorb by the body eventually and getting removed. And there's no liver damage to the normal liver tissue. High precision. So within the treated region, what you will see is that unlike outside treated region, you have intact cells. Inside the treated region, there's just no cellular components. They are all disrupted to acellular debris. And at the boundary, you can even see half of the cell, and the other half cell cut off, because what we are doing here is we generate cavitation microbubbles, and it is a threshold phenomenon. So when you have ultrasound pressure in the focal zone, above that threshold, you will generate bubbles and damage the cellular structure. And right outside, there's the ultrasound pressure is below the threshold, you have no damage. So the boundary is really sharp and the precision is very high. And third is real-time image guidance. So as I mentioned, ultrasonic image is used to guide the treatment in real time. So you can see the red outline, the red line is a circle is actually the clinician circled out the treatment region. Then the cross arrow indicates ultrasound focus. So during the treatment, the ultrasound focus is just scanned through this region where you can see the bright spot behind the red cross arrow because the bubbles actually appear as this bright spot, breast temperature changing spots on ultrasound imaging. So the clinician still can see what's going on even though they are not open the patient up, which helps us to really ensure high safety and high efficacy. Most patients wake up and then the first thing they ask is, have you done anything to me? Because most patients don't feel pain or no to minimal pain, and they get to go home right after procedure and have dinner at home and being able to return to normal activity the second day. So the very fast recovery. And very interestingly, the histotripsy also has this tissue selectivity feature because the critical structures such as major blood vessels and nerves and the bile ducts, those have higher mechanical strength and more resistant to histotripsy damage. So we are able to actually treat, including those structures within the treatment region, but being able to preserve actually the major blood vessels and nerves and the bile ducts. And this has allowed us to treat surgically inoperable location, for example, a tumor sandwiched between vessels without damaging the vessels. And lastly, immunostimulation. That was actually a surprising discovery later. We found that histotripsy treatment actually stimulates potent immune response, which is a long story, but it disrupts the cell membrane, help release the tumor antigen, and also cause immunological cell death, inflammation, and all those together mount into this really potent immune response in selected patients. So what we have seen is that treatment of one tumor actually resulting regression of off-target tumors. So in this patients, over a dozen of off-target tumors also regressed after treatment of the target tumor, which is actually not listed on this slide. So you can see 8 weeks later, the image shows that the tumor volume significantly reduced. So I started actually 2001, 25 years ago. So it's a long journey. And it was part of my PhD dissertation. And I discovered the histotripsy phenomenon in 2002. We published the first paper, so that was my first author paper in 2004, and quickly found out that there's critical things that scientific issues that need to be solved, including building specialized device for histotripsy and study the mechanism. In December 2009, I and my colleagues and I co-founded Histosonics. Now I'll tell you why I feel like we founded Histosonics a little too early, but there's a reason. And 2016 was the first clinical trial. This is actually was in prostate. And the trial results was mixed, and it was viewed as a failure by our venture investors. And Hysosonics almost died. We ran out of money, couldn't keep the lights on, and I will tell the story later too. But we switched direction to liver. And 2017 built the first human liver system, and 2019 was a big turning point. We conducted the first human trial on histotripsy cancer treatment. This is in liver tumors. And the phase 1 trial was the results were very good. And that really were able to convince investors to put in more money. And we started the multi-center clinical trial in 2021 called Hope for Liver Trial. And well, COVID hits. So also, it was a very difficult trial to do, but the results were good, and that led to the 2023 FDA approval on histotripsy non-invasive treatment of liver tumors. And 2024, we started a trial on renal tumors in the kidney. And actually, that has been pretty smooth, and the enrollment already complete, and we're just doing the follow-up right now. And December 2024, so this just last year, started a pancreatic tumor trial, and it has also been going great. To date, there are about 3,000 patients treated, mostly in the United States, but also in Europe and Asia. And as Dave mentioned, was recently, it was a private acquisition. So it wasn't it's a difference between private acquisition and a public, big public company just gobble you up. So anyway, so it was acquired for about $2.5 billion, and the Hysosonics is evaluated at $3 billion now. Okay, so my 25-year journey, and I'm going to try to talk about the big turning point along the way with lessons learned. Starting in 2001, and the reason I started this whole quest on histotripsy is because pediatric cardiologist Ahi Ludomirsky, I still remember him clearly. So in 2001, he came to my lab. I was a new PhD student. And he came to the lab, said, can you invent a non-invasive interventional technology that can perform the real surgery? And the reason he said the real surgery is because he was dealing with congenital heart disease patients, and they're kids, they're little babies. And he need to remove tissue from their heart and perform procedure, and the super invasive. And so his patients were so sick that they were dying on the surgical table. So he came in and asked, can you, I have access to radiation or radiofrequency ablation, even using ultrasound to do heating, but none of those interventional technology allow me to do the real surgery to physically, mechanically remove tissue. And I need something that can allow me to mechanically remove tissue like a real surgery, but non-invasively to save my patients. And I found that to be so inspiring. And after he gave his speech, I went to my advisor and asked to have that for my PhD dissertation. And apparently it was crazy. So it was a very new thing. And short story is I started doing experiments and literally going to slaughterhouse every week to get pig heart tissue, failed for the whole year. It was in 2002, because of a comments from a lab mate to challenge me to get a silent ultrasound therapy sequence, I came up with this special ultrasound sequence. I use microsecond ultrasound pulse to actually with extremely high pressure, and just to test it out. So you are seeing a video from more than 20 years ago, and this is what I saw when I actually did that experiment. And this is a piece of pig heart tissue in the water tank, and the ultrasound device was 10 centimeters away. And please look at here, and you will see that there's like smoke coming out, but it turned out this is not smoke. Those are tissue debris. They are submicron, so they're so small, so look like smoke in the water tank. And I was just seeing the tissue disappearing in front of my eyes, and a minute later, I saw this really well-defined perforation through the pig heart tissue, and that was the first time I observed hisotrypsy. And it turned out, and I will talk about the mechanism why why this happened. And also I thought I was dreaming the first time. I had to do 3 times to realize, to confirm it's actually real. So that's why you see like 3 holes on this pig heart tissue. If you're looking at the histology within the treated region, as I mentioned, it's just acellular debris because it's mechanical. So really it's liquefied the tissue to acellular debris within target region. So quickly after I discovered the hisotrypsy and present to the scientific community, I was encountered with skepticism and basically disbelief and saying that this is impossible for 2 reasons, and 2 good reasons. One is that the parameters I was quoting and I used for hisotrypsy experiments could not be produced in a repeated way by the existing commercial device at the time. So this is actually showing the amplifier I used back then. It was the most powerful amplifier at the time, 1,000 watts. So it's ENI N1000 amplifier. And within a month after my experiments, I actually had to go to my PhD mentor Charles Kane's office and told him that I have good news and bad news. The good news is I was able to repeat the experiments again, so it's real. The bad news is I just destroyed the most expensive piece of equipment in the lab. But it's not, I don't think it's my fault. And then we realized that even the best equipment, commercial equipment at the time, just was not sufficient to do hisotrypsy. So we had to make a decision to really design and develop our own device if we want to keep down the road of hisotrypsy. And that's what we did. So spent a long time to develop the really specialized device. And this is a show 20 years later, our 260 element hisotrypsy array. By the way, this is just one element. Now we have hundreds of elements. And this amplifier back then, A1000, has to plug in a 380 volts power plug. And now we have amplifier, this can fit on my palm and has 8 channels, and each channel is 4 times more powerful than this. But it really took a lot of technology development to get to the specialized device to enable hisotrypsy. And the second reason that it was deemed impossible is because the mechanism was not unknown, that nobody at the time believed this microbubble generation, the bubble generation, which is called cavitation. So ultrasonic cavitation was always viewed as random effects, so stochastic effects that couldn't be controlled, and it was only harmful. So we had to demonstrate that this cavitation can be controlled with special ultrasonic sequence, and there's a way to control it to do non-invasive surgery. So that took almost 10 years, and Charles, my PhD mentor, and my 2 PhD students, Adam, Eli, we, over that 3-year effort, found out that we can, if we use one cycle ultrasonic pulse, what we are dealing with is actually the existing gas pockets in our body, and they are nanometer in size. And they have very strong surface tension. But what we can do is with ultrasound, if we can overcome that surface tension, which is over 200 times the atmosphere. But if we can overcome that surface tension within microseconds, now we can reliably generate microbubbles and this is actually referred to the pressure threshold in water-based tissue, and most of our tissue water-based. And then we can actually really reliably and predictably generate microbubbles. And then this is a high-speed video that was taken with ultra-high-speed camera under the microscope, showing those black structural microbubbles and coalescing into one large bubble. And those white structures are breast cancer cells embedded in the gel. So really, when then we generate bubbles and the bubble expand, they squeeze the cell, and when the bubble collapse, it pulls cells apart. So it's this really high local mechanical strain and stress that eventually just mechanically disrupt the cells and causing the cell disruption. So after we figured those out, in 2009, this is a time where we just had a kind of early results on the mechanism, and we just figure out the specialized device. And so it's kind of still early stage. But however, the Culture Foundation, which is a foundation fund translational activity, and there was a Culture Foundation that funding activity within University of Michigan, BME, and they hired a consultant, Tom Davidson, and he was a very successful serial entrepreneur. So he came in and he was just looking at different technology, and he gave supposedly gave Culture Foundation an evaluation of different technology. And he really liked historypsy. So he came to our lab and really got inspired. And 2009, I just started my tenure track position as assistant professor at the University of Michigan biomedical engineering department. So I was very new, and I was working with Charles King, my PhD mentor still in the department at the time. So we were working together. But Tom came and said, I like this technology, and I will help you form a company. You guys will, I will be able to help you raise money from the venture capital, the VC investors. And he was able to raise $11 million for Series A funding, which was a really big deal for a startup company, especially for a company in Midwest. So this is Tom Davidson, and then Charles King here, and we have the other co-founders, and Jim Berchlina and Chris Gibbons are the one. They are kind of the first management team, and there's also Tim Hall and Will Roberts. So we are the scientific founders of the company. So Hisosonics was funded. And we thought it was a good opportunity that shouldn't be passed, and so we went along. But now thinking back, and with with what I know now, if I have to give other people advice, is don't form the company too early. The reason is we formed the company really early. We didn't have a human prototype or any sort of the data is especially no clinical data, and very like a preclinical data also is sort of lacking in a way. Therefore, our evaluation of the company was pretty low, and that led to, and also it's not de-risked enough. So that led to, the founders having small amount of stock share to begin with. Then you dilute it over the years, and also I think I was particularly naive. I don't think any of my scientific co-founders really knew much about business either. So we were not savvy enough to be able to participate in negotiation of licensing term and negotiate a good licensing term. So that's actually important that for anybody found a company, being able to be part of that licensing term negotiation with your tech transfer office to form this company is something definitely need to educate yourself and look into that before just go ahead and sign the paperwork. But for us at the time, we thought this is a too good opportunity to miss, so we took opportunity. So it took 6 years to start the human trial because we had to also build the human prototype device. So Will Roberts is a urologist, so we work with him really closely at the early beginning, and he identified benign prostatic hyperplasia to be the first indication of historypsy, and which is enlarged prostate, right? And you have to go in to really just remove the prostate. We thought, liquefy the prostate, and then the patient can urinate it out. Then you you solve the symptoms and your treatment is done. So it seemed to be pretty straightforward and a great application. And that's why we went with it. We did all the experiments in this preclinical animal model, which is a canine prostate, and the results were just really amazing from the the canine prostate experiments. So we started a clinical trial, treated 25 patients with BPH, and was able to generate cavitation and observe on transrectal ultrasound imaging in all patients. And all patients also had a symptom relief, although it's only up to kind of 6 months you have symptom relief, but they all went back down in a year, all back to the baseline. And there wasn't any evidence of tissue liquefaction of the prostate as we actually saw in our preclinical experiments. And if you think about phase 1 trial, I think the phase 1 trial, if this considered a phase 1 trial, it's actually not bad, right? Because we demonstrated safety. The safety profile was really good. And also patient had no to minimal pain. And there's some symptom relief. But however, because there was no tissue liquefaction evidence, the VC thought that the efficacy data considered poor, and at the time, the company spent up all the Series A funding money already, funding dried out. So the investor said, we think it's a failure. And then what's next? So the lessons learned here, that, and I think I have talked to quite a few startup companies that backed by VC, and looking at the phase 1 trial, even though phase 1 trial is supposedly designed to test the safety, but the VC investors typically have expectation to see some efficacy data, and so they have their expectation. And if you are not meeting those expectations, then you're really, have a high likelihood that they just don't want to continue to invest. And for us, the reason we found out years later, the reason we didn't see the efficacy data as good as we saw in our animal model is because our animal model was not an accurate model of the human patient. So we were using young canine prostate, where it turned out the human BPH patient prostate is a lot more fibrous and takes a lot higher dose compared to the animal experiments. And we didn't know at the time, so we were way underdosing. But we didn't know that, and also we ran out of time and money to do another trial on this to fine-tune it. And what happened is that at the time, actually 2 years prior to this kind of failed trial, my lab had been already studying liver tumors for a while. So my lab started investigating liver tumor treatment with histotripsy in 2010. So we had good data. Johnson & Johnson was interested in liver cancer treatment because it's aligned with one of their main directions. So they came in. We were able to have a conversation with them, and his Sonics board, the chair, was able to engage them for quite a while, and were able to bring them in to my lab to look at the demo and look at the to get a demo and to ask questions and to really just figure out if they would be interested in this. And I still remember it because when Johnson & Johnson, their team came in, they had their leadership, VPs, and they have their surgeons, and they had their technical people. It was a large team. And it was the day right before that, I was 9 months pregnant with my second child. And the day before that, I had a gallbladder stone episode and was sent to ER. And I was in the ER, and they were thinking that they wanted trying to get me to deliver a baby. I told them, I was like, no, it's not happening. I need to go back to the lab. I need to do this demo. I need to give this presentation. It's extremely important. And I was able to convince the clinician to let me go. And I was, went to the lab the second day. gave a good presentation. The demo went really well. Even though I could barely walk, but I was able to convince Johnson & Johnson at the time that when his Sonics, this BPH trial didn't go well, they actually came in. They put in not a huge amount of money, but they did put in some money. And this was able to really boost the confidence in other investors because Johnson & Johnson is such a, big company in the field. So other investors are willing to put in some money too. But the condition is we switch to liver tumors. So we decided to switch to liver tumors and also change the management team. And that was also a time where there's a new management team came in. And this new management team, they were very experienced with, especially liver tumor field. They came from microwave ablation of liver tumors and had experience with taking the startup company to FDA approval, to even a U.S. market leader, and to IPO and the whole thing. So they need. Before when we were trying to do the BPH thing, the scientific founders was very much, engaged and part of the decision-making process. But after this, when we decided to switch to liver tumors, it was at a time point as that we, every scientific founder feel like you have a technology or you have this, science you do is your baby. But at some time point, when you found a company, you kind of have to decide that do you want to keep the control? And at some time point, you should just go over to the company, or are you, willing to lose control, but still get involved, but let someone kind of professional management team to make the business decision at least. So that there was a turning point at that time, and also part of the reason was that there was a time that scientific founders and management team were kind of at odds, where there was some disagreement. So this is a time where some people decide scientific team decide, maybe this is not for me to continue. And I decided to stay, but not having any control in the business making part. But I want to keep working on technology and making sure technology is okay and going forward. My lab did a lot of work in the liver tumor side, and I think that's not the purpose of this talk, so I'm not going to go into that deep. But I do want to show, this one slide where we actually did an experiment, lots of cohorts experiment. This one I think is very cool, which is this is an orthotopic rat liver tumor model. So you can see that it's a highly aggressive, this is HCC model, hepatocellular carcinoma. Without the treatment, the tumor really just metastasized all over the liver, and then we have to euthanize all the rats in the controlled untreated group within 2 to 3 weeks. With histotripsy, we started treatment about 1 week after the tumor inoculation. And we treated about 50 to 75% of tumor. So this is where they start to kind of spread, but not really, hasn't spread much yet. And Great. Okay. 好的，好的。Okay. Okay. I'm sorry. Okay. All right. But in the treated cohort, we had that for that batch of 11 rats, 9 out of 11 rats, we actually purposely treated part of tumor because there was a concern that as a mechanical approach where we disrupt tumor cells, are we going to actually release tumor cells that can then result in increased metastasis? So we purposely did that experiments where we treat part of tumor, then wait to see what happened. And it was surprising but great results that 9 out of 11 treated rats actually had a complete tumor regression. And later we found, learned that it was due to the immune response. And 2 of them had also increased survival. So we had a long kind of and really a great amount of preclinical data that led to one is this Edison historypsy system, as I mentioned, that's ultrasound image guided and robot arm assisted. And then also the start of the Hope for Liver trial, that's multi-center trial on the liver cancer patients. And this was done during the COVID, so it wasn't really hard to recruit patients and a difficult trial to do. And I don't want to go into the detail too much, but it was showed that the safety, technical success was good, meaning that 90. So the tumor that you targeted, the intent to target, so the clinician intent to target tumor, and then was able to treat the target tumor and confirm on the imaging. So that's the technical success criteria. So then technical success criteria was really high. Technical success was more than 95%. And this is actually really, I thought it was great because you have to realize those clinicians are really, they are new users. They never used historypsy before. They got trained and then quickly have to then do the trial. So considering that they are, mostly new users, and I think this reach is actually quite high. And showing historypsy can be learned pretty easily. And also the safety profile is really good. There were 3 incidents of adverse events, but adverse events. But it was considered later that there were 2 minor ones that was resolved with antibiotics or just a painkiller, and there's one had a hepatic failure, but it's not related to the treatment. It was just really end-stage patients. And they'll have 1 year follow-up data showed 90% tumor regression. So all those led to the FDA approval in October 2023 for non-invasive treatment, historypsy treatment of liver tumors and using the Edison system. And I will mention this later. It was approved under the de novo pathway, which is something that really the regulatory pathway is actually really important, and something can be very I will go through this actually. So the lessons learned of that process is that first, the right regulatory pathway is really critical. So for regenerative medicine, I'm not an expert in regenerative medicine. The IND side, actually, I'm not familiar with. So I'm more, kind of, my knowledge comes from the IDE side, which is exempt device, investigational device exemption. So for the IDE side, you can have 3 pathways. One is the easiest one is a 510K. So 510K is where you have already present, so predicate device that's already in the market, FDA approved, and you just are making incremental improvement. And in that case, you can cite, for example, you're doing an ultrasound imaging device, and there are other ultrasound imaging devices out there already. So you can say, I'm going, with a 510K, and that pathway is relatively easy. Or clinical trial is very small, and you don't have to really, the requirements for follow-up data and all that would be low. PMA is on the other hand, which is very new technology, so you don't have any predicate, and you have to therefore get big trials, and you have to the requirement is high, and but when you get it, and then you can really make claims that other people cannot claim. De novo is in the middle, and with historypsy at the beginning, everybody thought that maybe we have to go to PMA because it's new, but the management team, Hysonics, the regulatory team, just very experienced. So they were able to pivot to this de novo pathway with a breakthrough device track. And this really was able to allow them to kind of fast forward the process and reduce the requirements on the FDA side, and being able to reduce the number of patients required for the clinical trial and make it more achievable, so that we can save costs and money. And even that is a huge amount of money and time to be able to get through this. And second is that especially for a new thing, right? Whether you have new technology or new drugs, or new like regenerative medicine thing, there is a possibility that FDA just doesn't have the knowledge to, as compared to some mature technology or mature kind of a drug pathway or device, so that or maybe existing standard does not directly apply. That you really need to educate FDA to give you the right evaluation. And in our case, that was the case, and there wasn't any existing standards at FDA side to be able to allow them to evaluate histotripsy in the right way. Therefore, we had to, Hysonics and actually their regulatory team did a great job to really have a constant, very, very open communication channel with FDA to educate them. I would say that, I know FDA had a kind of a, their social reputation, but in my experience, that they actually really willing to talk to you, and for new things, and willing to, trying to listen and trying to come up with the right evaluation. So I thought that especially for new technology or new drugs, that communication with the FDA actually is very helpful. My third point, I as a lesson learned here, and I really don't want you to take it the wrong way, as I said, trying to set a low bar for the clinical trial success criteria. And the reason I said that is because I found that when we were, when I was talking to a lot of my colleagues, when you write grants, right, you go for the big impact, right, high impact, and you want to claim, the great, efficacy data, the improvement and all that, and that's important for the scientists like us. However, going through the FDA process, there's just so many uncertainties when you go into the clinical trial. So there's a lot of things you cannot anticipate. And for FDA, the really important thing is making sure you have a really excellent safety profile. That's important, that your, any benefit you can provide, right? So you kind of have balance. You are being able to be safe, then you can provide some benefits. And you set a kind of criteria going in. And the reason I said here is that I think when you are designing a clinical trial and a protocol, just keep in mind that don't trying to overclaim and set the bar too high for yourself, knowing that in the human trial with a lot of complicated things going on, that you may or may not be able to achieve your ideal expectation. But you want to go to the FDA, because once you get the FDA, if people start using it, you get clinical data, you learn actually so much more even after the trial, right? The post FDA approval, where you have the real clinical use, that's where you learn tremendously. So the goal is to just get through, right? So if you can get through it, you will continue to learn clinical knowledge after the FDA approval. Then the fourth point was also really important. It's also very important, which I I did not know before. I thought, before I said, oh, let's go through FDA. If we get FDA, that will be enough. But it turned out that FDA approval, once you get it, you actually go nowhere if you don't have reimbursement. So reimbursement with medical insurance is critically important. I can't emphasize this enough, that in order to get clinical adoption, clinical use, like nobody, you don't have reimbursement code, you don't have that, through the medical insurance, Medicare, right, major insurance company, at least for the medical device, that you wouldn't be able to sell any machine at all. And the reimbursement need to be considered in the clinical trial design so that you can produce that data that you can use towards the later reimbursement. Then lastly, it's I think you probably know it, but I think you need to set the right expectation. Even you get FDA approval, it doesn't mean widespread clinical adoption. The path for clinical adoption for any new device or drug is very, very long. So it took me 25 years to go through from an invention to FDA approval to get reimbursement so we can sell machine and have some clinical use and with 3,000 patients treated. But we're still far away from wide clinical adoption. That is a road that need many things to happen. And I do want to mention one thing. I think is very helpful to have a clinical key opinion leader as a champion that can help you to, you have to keep generating clinical data, but have that voice to help you within the clinical community. And also just, overall that setting up the right kind of scenario of clinical use, right? So when you go out, like your first indication is important. How you want to see the clinical use of for that initial indication, and how people talk about it, how the message get delivered is important. So it's a very long road, but I think from my personal experience, it's extremely rewarding when you actually see patients getting treated and really saving people's life, change their life. And I can't really put any really, like, it's just extremely rewarding. I can't imagine anything can compete with that. So it's still really worth doing. So at the end, I do want to leave just a couple of minutes, but I just want to acknowledge my team for really big team effort. It's not really, I'm only listing kind of my research team, but there's all the a lot of business side team that's really have been working together. I used to say that it takes a village to really do this work, but what's really happening is it's taking an army to do the clinical translation work, not just the research side of people. I have my clinical collaborators. Of course, the federal funding has been extremely important for development of new technologies, and then also the whole Hysosonics management team. There are all those clinical users and the patients too. Really, just a whole army of people for any new kind of technology or new medicine to be able to get to that clinical adoption point. So with that, I thank you, and hopefully I'm trying to turn on my video again. Still doesn't work. Oh, now it works. Okay.
Okay, thanks, Jen. I think your last slide was spot on, because all of those bullets were kind of agnostic to technology, and we've really stressed to our project teams each of those bullets at one point or another. So thank you. Dr. Chen, you're welcome to come up here, and we could just open the floor up for questions. I know there were a couple of questions I wanted to get in after Dr. Chen's talk, so I'll see if those people want to ask those questions first. Did you have a question, Steve, for Dr. Chen?
because that's what I am, true. Did you go down in your FTE? Did you go, because they say, for instance, within STTR and SBIR, right? Then you have to decide who your CEO is going to be. If you're 100% at the university, you're making a decision right away about whether or not you're going to be the CEO for the, which means you're going to have to come down in your FTE or trust somebody in your lab to do it, which is what I did, which I'm doing, because there are different ways to do it. But then it's a jealous mistress. As you are doing that, it pulls you more and more there. So how did you negotiate that? And I don't mean negotiate with money, but you yourself about being this clinician and this researcher that is in the university and then going off onto the business as well, especially when you're raising money, they want you to be as a founder potentially full-time there. So you have these two jealous mistresses pulling back and forth. How did you deal with that? And did you stay 100% at the university or did you ultimately go over to the other side?
No, I've always stayed at the university, but my pay though is as a clinician, and so our pay is basically just eat what you kill, and so the cases I do are what I get paid for. But in terms of like the SBIR, STTR grants, so I would be the PI for the STTR grant, and then I would subcontract to my laboratory, and those people would be basically one of them would be a PI for the subcontract. And so I've never taken salary as a PI for the grant, basically. So I have a % effort, but then it's all basically placed with the company. Now, in terms of this, it's basically this whole thing with conflict of interest. Conflict of interest in terms of what is the university as a university employee and what is the company. So I was a lot happier when the company was not public because nobody paid any attention to me. And then the company went public. And all of a sudden, all the USC lawyers are like asking, compliance and all that stuff. And so, like for instance, in my laboratory, I was fairly loose in terms of like who was working on NEONC projects and who was working on USC projects. And so I just recently, for instance, the lawyers got all on top of me, like, your USC employees, what are they doing that's NEONC and what are they doing that's grant related? And so what I did was that I took everybody's grant support and said, okay, so this is your grant support, this is your FTE from all the grants combined that you have grant support. But it was not quite enough for them to make the salary that they wanted to make. And so what I did was that I then had them do a work for hire. And so the company was then paying them for their work. And that brought their salary up to the desired amount. So everybody then was a USC employee getting USC benefits. They were working on work that was grant supported, and then they were working on what the company contracted them to be doing. So.
Yeah, Pam.
Have you ever been audited?
I will be.
Because
right now I'm still poor because all my shares are restricted.
Well, my understanding was that once you received a phase 2 STTR,
Yeah.
that you will be audited or would be audited, and they would go back to your phase one, which we right now have a phase one with TTR, and to be honest, are navigating it as best we can as far as everything that we're supposed to be doing, but I'm not really sure that we're doing it correctly, reading a lot of self-help books on STTR phase one and phase twos, but my university is really nervous about every single thing that we're doing because they're worried about being audited. I mean, that's really what they're worried about. So I'm really surprised that I would hold your breath. No, I'm only kidding, but best of luck with it all. But it's really complicated, and not ignorance is no excuse for what you're doing. And
I know, that's why.
for what any of us are doing.
Believe me, I've got enough lawyers. I've got a big bullseye on my back right now.
Yeah, yeah, I'm familiar with that, and
like
at a
I-
much smaller scale than yours.
Like I said, that I think I was a lot happier if I was nobody paying attention to me.
Okay, I think Steve had a question.
My understanding is that you are audited only if you receive like over $1 million, or there's some like threshold if you receive over a certain amount per year. I'm not sure you're automatically automated with, or with a phase two audit. Yeah, but it's per year, so it depends on how long your grant, you know, how long, because it's usually a 2-year grant, so, yeah, and the funding amounts have gone up, so I'm not sure if the thresholds have gone up, but
So, like,
that
a
was
lot-
how we stayed away from it.
Yeah, so a lot of our, like some of our grants are what's called straight to phase two. And those straight to phase two are like 2.1 million over 2 years.
Well, I wanted to start off by thanking both speakers for really engaging presentations. Dr. Zhu, one of our 4 sons, took a class with you and was very inspired by your lectures and your enthusiasm. He's now with Epic in Wisconsin. His name is Austin Bloomberg, and I don't know if you remember him, but
Yes, I do. I do.
oh,
Wow.
wonderful.
That's
Well,
great to hear.
he gives his regards. And Dr. Chen, your presentation, I'm a chemist by trade, so immediately started wondering, what is the darn mechanism here? I looked at the molecule, I had to look it up. It's a non-conjugated double bond system. It must be binding to cell sites and causing apoptosis. But can you allude a bit on the mechanism and why it works for some of these cancers? I think you kind of indicated maybe not for glioblastoma, but more for astrocytoma and meningioma.
So basically, as I mentioned, that the para-alcohol was initially worked on as a cell cycle inhibitor. At that time, they also found it to have some effect on the RAS oncogene. But for us, when we worked it out in the laboratory, what it does is that it essentially permealizes the cell membrane, and so it induces ER stress, and then ER stress into apoptosis. And that's what I think is the main mechanism by which it works. And so, when we start looking at it as a mechanism, we really feel that this whole idea of inducing stress in the cell and leading to apoptosis is probably what happens. Now, in a rapidly growing cell, we found that that's pretty much the mechanism, that ER stress apoptosis. And in a slower growing cell, like a meningioma that we have in one of our trials, we find that it basically goes through autophagy first, and then autophagy to apoptosis. But the whole idea of everything that we're doing is that we have a treatment that is non-invasive, but it still is for the most part, cytostatic initially. And then over time, it can become cytotoxic and end up killing the cell tumor.
Well, maybe you can both travel together to, where did you say? Was it not Qatar, but, and you can say that together you can cure all cancers that are ever.
Dr. Zhu, can I ask you a question? This is from clinical adoption standpoint, and I don't know if you've thought about this, but as a surgeon, I'm wondering, when you talk about your champions, are your plans to go to the surgeons to use this? Because if you're not, and you're thinking going to hepatologists or you're going to radiologists to do it, the surgeons are not going to like you very much. And I know this sounds crazy, but it's going to be a really important decision about who you think you're going to give this technology to. Because if the surgeons feel like it is an attack to them and they're going to lose market share, they're not going to be very happy, and you'll be fighting against them that are there. So I'm wondering, how is your business thinking about this? Is it going to the people that would normally resect and maybe now can go to unresectable in some way, or are you going to give it to a different specialty and then they're going to take it forward? And I wonder about the political battle. As a surgeon, I could just see it happening, and it's going to be a pretty important decision for you to make as a company. I was wondering how you're going to approach that.
We love surgeons. We have majority of our users right now are surgeons. So it's more than 50%. There's another big is interventional radiologist. So we have, then there's a very small fraction of radiation oncologists. So we do not want to exclude anybody, but surgeons are our biggest user right now. And in the liver, because there's still IR, interventional radiologists perform the ablation procedure, so that, and they are familiar with imaging. But when we extend to other organs, thinking about pancreatic tumors and also going out prostate, breast, and thyroid, and the brain, and all that, and those surgeons probably make even more sense. So we're definitely engaging surgeons for sure. But we don't want to exclude just exclusively only oriented towards surgeons. We do want to include other specialties, whatever it makes sense clinically. But yeah, no, our champions are actually there are a lot of surgeons as our champions. We have engaged a lot of surgical departments and also presented at the surgical conferences. Definitely love surgeons.
Can I ask a quick question of Shan? I love this work that you're doing, and I was thinking about the diversity of applications, and you talked a little bit about tumor types and the levels, the sizes of tumors that you can be actually acting on, and also think about the technology in the future. So in terms of product, moving forward with your product and with your application, what do you see as the next phase for this method for your, are you going smaller tumor size? What's the pipeline that you're currently working on to really create even a more beneficial therapy?
Yes. So we so just to give you an example as kind of a clinical experience in terms of the size of the tumors and the number of the tumors that can be treated. So currently our kind of record so far is so this is just one session treatment. So the patient come in, unlike radiation, you have fractional treatment. They just come in one session and they get a treatment. Some may get repeated treatment if needed, but the record right now is treating up to 10 tumors in one session. And that's a surgeon. He did a 4-hour procedure to treat 10 tumors, and we were just shocked when I heard it. But he said, I can do 8-hour surgery to remove that 10 tumors. I'll do a 4-hour procedure with histotripsy. So, and the largest tumor that have been treated so far is 10 centimeter in size. So it's so really there's a reason, not a real limit in terms of size of tumor and number of tumor nodules that can be treated. It's more just about the treatment time. And in terms of different organs that we're looking at, we really just trying to expand to where non-invasive surgery can really benefit patients. As I indicated, renal tumor, pancreatic tumor, already clinical trials ongoing. We anticipate getting FDA approval for those in a year or two. As I mentioned, that in the ones in the pipeline include already in the short term include prostate, thyroid, breast, and we actually going to start even the brain trial in probably 2 years also. So the pipeline created is more about not only just the technology on our side, but also on what are the clinical indications that based on our investigation think can be accepted by clinical community, and also from the investor point of view, what can make money, right? So, so they're looking at large things.
People
large
buying
markets.
into it is important too, but you've got that already. So we're always thinking about how can you get people into invest, and you've got the success. So I assume more money is just going to fall from the sky as usual. Thanks.
Yeah, thank you. Yeah, but finding investor is definitely there's an art in it. I will have to say, how you get funding. Oh, sorry, I'm looking at the chat. There's a question in the chat, but yeah, so definitely getting the investor when you go in. I think again, like from academic point of view, we talk about high impact and all that, but I think you guys are already seem to be very very experienced already, but really looking at market acceptance, right? How you can enter clinically. And how you can spread it out. You have to have a very solid business plan, and then you really have to have a good story to sell. And the people with track record, the business manager with track record can really help substantially. So yeah, the money doesn't fall from the sky, but there are ways and art to raise money that can make it more attainable. So.
Let's come over here with a question, and Vivi, I think there's a question in the chat, if you could read that.
Yeah, Dr. Chan, the first question is, how come you went to public? Could you just continue on the privately raising the money? And then the second question that I have for you is that would it be better, maybe the patients, I mean, this is kind of maybe a stupid question, but would it be better the patients laying down in a spine position while administering this, the nebulizer, so that it could get to the nerve better with that position?
Okay, so yeah, so that's I'll answer the second question first. That's an interesting question, and when we do the intranasal delivery for the mice, that is the position that they're in. But I've never tried it lying down for the patient, mainly because when you do this delivery, it's not a clean delivery, unfortunately, or essentially all of it just goes up to the nose. You get a lot of it going down to the throat, and so one of the things that I had thought about was that I didn't want the patient to feel like they were choking when you're taking this, but I understand what you're coming from an anatomical standpoint of getting better coverage. Now, in terms of going public versus private, it's a lot easier to be private in terms of what you can do. If you can generate enough cash to last you, and then to be able to generate enough, eventually generate some revenue so that you can have some profit staying private. The main problem is that biotech basically runs on science and money. And like we burn about $500 to $1 million a month trying to run 4 clinical trials. And so unless I was, I don't know, unless I had some tremendous technology like that somebody was going to dump a whole amount of money to me, it is still very hard to raise money on a private basis. Public, it is easier to raise money public. And like, for instance, we were placed into the Russell Index. And just because we're in the Russell Index, in the microcap Russell Index, we automatically got institutional investors because they have to, anybody that wants to be part of Russell has to buy part of our company. And so there are things that can be done in the public market that's much easier for us. And then the last thing is that, my shareholders, the people that, my legacy shareholders, they've been with me for like 13 years. They haven't seen a cent in terms of return for their investment. And then when you go public, they can actually realize some of that investment.
Yeah, I think there's a question in the chat.
Yes, there's a question from Kay Fuller to Dr. Xu. Are you and your team studying how the immune response is occurring?
Yeah, I actually responded to Kay in the chat that yes, we are. We are studying the mechanism behind the immune response, and also we're studying combining histotripsy with immunodrugs to enhance immunotherapy. So we are looking at results that it seems like histotripsy is stimulating immune response even in poorly immunogenic diseases where patients are not responsive to immune drugs. So we can synthesize those the immune response such that then in a way turn the cold tumors hot. So that is definitely another big direction that we're taking to combining histotripsy with immune drugs, and hope to expand non-invasive focal treatment to kind of systemic treatment in the future. So definitely a direction of investigation. Yeah, thank you.
Okay, we're right at 5 o'clock. I'll take one last question. Yes.
Yeah, this one for Dr. Xu. Again, great presentation and great technology. What are the risks for metastatic movement while this procedure is happening? Are you following those as well?
Yeah, we're following. So it's hard to say. I mean, so far the preclinical study doesn't suggest any risk of increasing metastasis. It is, if anything, it's reducing the metastasis. But different clinicians have different kind of way of care for their students, care for their patients. There are clinicians are stopping chemotherapy for their patients when they're doing histotripsy treatment, even though it's not required, because they have seen those patients get better immune response. But their clinicians just doing histotripsy treatment while still keeping the patients on chemo. So it's hard to tell right now what is the best clinical practice. So I don't think there's really a risk, but it's kind of muddy field right now because you have we're dealing with a lot of patients at very late stage with metastasis everywhere, and it's not exactly clear yet what is the best protocol to care for those patients, and the clinicians are just in their own way making the best judgment they can make right now. So we're still learning.
Okay, thank you, Dr. Xu, Dr. Chen, for a great session. Thanks to you all.